Welcome back to the gastrointestinal physiology. Um, Today I will be talking about two of the special functions of the GI tract. I will be talking about digestion and I will be talking about absorption. Uh, Now, just a little reminder, we have divided the topic of GI physiology into general physiology and special physiology. General physiology, we talked about a definition, structure, and control systems of the gastrointestinal tract. And in the special physiology part, we talked about special functions of the gut, such as motility and also secretion. This lecture, I will be talking about two special functions for the gastrointestinal tract, and these functions are digestion and absorption. And the reason why I put these two together is that I want to make a story out of them. So what I want to do is once I finish talking about one of the nutrients, say carbohydrate, I want to continue talking about how will we absorb carbohydrates inside in the enterocyte and then to the bloodstream. So that's why I want to talk about the two functions together. We digest it first and then we utilize it second for the three nutrients: carbohydrates, proteins, and fat. Again, the reason for choosing two of the functions together is basically we want to connect them together. We want to make a story out of them. I don't want you to forget about, one of the functions when I talk about the other. I want the two things to be connected. We digest and then we take it inside the cell and into the bloodstream or to the lacteal or the lymphatic tissue. So that's the reason why the two are together. So the first thing is that digestion is basically reducing the size of the ingested material into a acceptable or reasonable size in order for the body, the different systems, to utilize it. If you eat, for example, a piece of steak, the cell is not going to be able to absorb that piece of steak. However, what happens is that when you digest and break and decrease the size of that piece of steak into a pretty reasonable size, that is when the body basically absorb it and utilize it. The body gets the most benefit out of it. Again, first you have to give it to the body in an acceptable size, in a reasonable size. The digestion process happens all over the gut. There is digestion, different types of digestion, all over the gut, all the way from the mouth to the large intestine. Different degrees of digestion, but there is some degree of digestion along the whole gastrointestinal tract. So digestion is decreasing the size. Absorption, on the other hand, is passing the nutrients, again, the acceptable size of those nutrients, carbohydrates, fat, and protein, into the enterocyte first, and then into the either bloodstream or the lacteal or the lymphatic stream. So two paths. First, you're going to get it from the lumen of the intestine to the inside of the enterocyte. One. Two is to get it from the enterocyte into the either the blood circulation through the portal vein or through the lymphatic circulation through the lacteal. That's how it's going to get to the general circulation. And this happens, unlike the digestion, which happens all over the gastrointestinal tract, absorption mostly happens in the small intestine. This is a picture that shows the couple of villi in the intestine, and it also shows the different enterocytes that basically line up on this villi, and also the microvilli that protrudes from these enterocytes. As I mentioned in the very first lecture in gastrointestinal physiology, these microvilli work as sensors to the lumen of the gastrointestinal tract. They sense the internal environment of the gut lumen. Inside these villi, there are three types of vessels. Well, two vessels and a nerve. So we have an arteriole, we have a vein or a venule, which is smaller than a vein, venule, and then we have a nerve, of course, enteric nerves, and vagal afferent. If you remember the talk on vagal afferents, we have one vagal afferent that reaches the villi, intravillous arbors, if you remember that. And the other thing that this villi contain is a lacteal or a lymphatic vessel. It's called a lacteal. Now, the upper cells in these villi are very, very active compared to the bottom cells. They're very active and they can regenerate very quickly. The bottom cells are considered a defense line for the gastrointestinal tract. Any bacteria, any parasite, any toxin, things like that might harm the gastrointestinal system. The bottom cells usually take care of them. Now, one very important thing that I would like for all of you to remember is that there is an age-related enzymatic activity. What does that mean? It means that the activity of the digestive enzymes is decreased with age. So the older we get, the less active these enzymes become. This is a very important concept that we would like to remember. So that might also explain why older individuals or animals might have more digestive problems compared to younger individuals, both humans and animals. So we need to remember that. The reason for these villi and crypts is basically they increase the surface area. When you open them up, for example, they can come up to about 400 square meter. That's a huge, huge space, which means it is sufficient for digestion of food. This is another picture of the villi and the crypts and the structure of the wall of the gastrointestinal tract, which you've seen before. But this is to show you the upper cells, which are enzymatically active, versus the lower cells, which are considered to be a more defense oriented, or they are concerned with considered as a defense mechanism for the gastrointestinal tract. And also you can see the lymphatic vessel, which we call lacteal. The lymphatic capillary or lymphatic vessel, we call that lacteal. We'll talk about that in a second. Now, we will talk about the different types of digestion first. So after we looked at the type of cells and how they are organized in these villi and the crypts and the type of these cells, now we want to see how does digestion take place. Well, there are two types of digestion. There is a luminal digestion. We call also this type of digestion, we call it cavital, from cavity, the word cavity. So it's the lumen of the gut, cavity of the gut, luminar or cavital. The other type of digestion is a membranous or contact, which I'll explain it, of course, in a second. But before we go there, let me just say that these two types of digestion, one happens or occurs in the lumen of the gut, that's why we call it luminal, and the other one, the nutrient must be bound to the membrane of the enterocyte in order for it to be digested further, to be able to get to the enterocyte for the absorption process. So first we have to decrease the size in the lumen, the size of food, the size of that piece of steak that we've talked about in the lumen, and then after that, the tiny material, I'll talk about that in details later, the products of this luminal digestion will be attached to the membrane of the enterocyte, and that's when it will activate as another set of digestive enzymes to break it even more to be able to get it to the inside of the enterocyte. So let's talk about that. Luminal digestion. Okay. Luminal digestion, as I mentioned earlier, occurs in the lumen of the gastrointestinal tract, which means any lumen in the gastrointestinal tract contains digestive enzymes. And I said earlier that digestion occurs throughout the gastrointestinal tract. Absorption, on the other hand, is mostly in the small intestines. So let's talk about the lumen first, the luminal digestion. Where are the lumens in the gastrointestinal tract? Well, the oral cavity is a lumen. It has saliva, it has lipases, it has amylases, etc. The stomach is a lumen. It has hydrochloric acid, and it has pepsin, which is the result of pepsinogen activation. Okay, and the third one, we have intestinal and pancreatic digestion, or the lumen that happens in the lumen of the intestine. So we have three sites where we have digestion in the lumen of the gastrointestinal tract. In the oral cavity, with the saliva and lipases and amylases. We have it in the stomach, and we have hydrochloric acid and pepsin. And of course, the most important one is the intestinal, because you have tons of pancreatic enzymes that are very, very, very important for the digestion, as we will mention next. Now let's again. So these are all general. We're talking general before we talk about the specific nutrients. Before I talk about how we're going to digest carbohydrates, before how we're going to digest fat and protein. So these are all general. Now the membrane digestion. The membrane digestion is basically occur by enzymatic or hydrolytic enzymes. Hydrolytic enzymes. Enzymes that hydrolyze the nutrients, make them water soluble. Okay, and all of these enzymes, the membrane enzymes, are bound to the membrane, the apical membrane of the enterocyte. That's why it's called membranous digestion. Again, we have two types of digestion: cavital or luminal, that's in the lumen, and we had three lumens oral cavity, stomach, and intestine. And we have membranous. And membranous, it's basically a membrane that is bound to the surface of the enterocyte. We call that the apical membrane of the enterocyte. This is a picture that shows basically the different layers all the way from the lumen on the top of the picture, the lumen of the gut, and you have an unstirred layer. Then you have a microvilli. Of course, after that microvilli comes the tight junctions and the enterocytes, which are the cells, basically the cells that lines or the epithelial cells of the intestines. And we will talk about these layers when we get to the nutrients, basically. But we can see, at least from this picture, that the material in the lumen has to go through diffusion in order for it to get into the enterocyte, and then from the enterocyte into the capillary, which are basically located in the basement membrane. So this picture basically shows you the type of layers that nutrients must pass to get into the enterocyte and then to the capillary. How are we going to transport this? Again, all of this is general now. We did not talk about special digestion of carbohydrates or proteins or fat. All of this is basically general concepts. So how does the material gets from the lumen of the gut into the inside of the enterocyte, and from the inside of the enterocyte to the blood vessel or to the lacteal, so it can go to the rest of the body, so the body can utilize it, can use it? There are a number of methods. We have simple diffusion, and simple diffusion is basically through the holes, if you will, the pores of lipid domains in the membrane of the enterocyte that the material or the nutrients gets through. Simple diffusion, not too much energy spent, no special carriers required, etc. So it's a simple diffusion from one side to the other. Active transport actually requires a energy. You have to spend energy to actually push that nutrient across that apical membrane of the enterocyte to get it inside to the enterocyte from the lumen of the gut. That's active transport. Facilitated diffusion, it's basically some special type of diffusion that requires carriers. You have to have special molecules that grab to certain nutrients and take them by hand to get them from the lumen of the gut to the inside of the enterocyte. That's very important. So special molecules that needs to carry the nutrients across the apical membrane of the enterocyte. And the final way for transport is pinocytosis, which is basically for larger molecules. The cell basically surrounds the molecule and tries to swallow it, just like a bacteria, just like a method whereby you change the nutrient inside the cell and not outside. This happens with antibodies, antigen reactions, and things like that. So if you have a bigger molecule, not necessarily a nutrient or something that the body's gonna utilize for nutritional values. As I mentioned earlier, active transport requires, of course, energy and special carriers. We have to have special carriers. If we have secondary active transport, then we don't need any energy or carriers. So it's basically two types, primary and secondary. But the most important thing to understand is that when we say the word active, that means we need energy and we need special carriers for that. Now, let's talk about each of the nutrients separately. So we have three different nutrients. It's well known: carbohydrates, proteins, and fats. We don't have any other thing. and any nutrient that we consume falls into these three categories. So what I'm going to do is I'm going to say first I'm going to take carbohydrates. I'm going to tell you how it's being digested both in the lumen and in the membrane. Remember, we have two types of digestion, luminal and membranous. We have to talk about these two. And then I will tell you how we're going to absorb the end product of carbohydrate digestion inside to the inside of the enterocyte, and then from the inside of the enterocyte to the blood vessel to take it to the rest of the body. And the same thing I'm going to do for proteins, and same thing I'm going to do for fat. So let's start with carbohydrates now. The end product for carbohydrates digestion is glucose, fructose, and galactose. This is the end. Any carbohydrate that you consume, no matter what type of ice cream you eat, or type of bread you eat, or type of any carb that you consume, you or the animal, both a small and large animal, the end product is going to be glucose, fructose, and galactose. That's all. So the question, how is we are going to get to this point when we eat any carbohydrate, bread, wheat, ice cream, you name it, sugars. That's what I mean by ice cream, sugars, not the fat or the protein in the ice cream. That we'll talk about it later. But I'm talking about the carbohydrate component. Okay, so this little table will basically summarize to you how is carbohydrates being digested. So on the left-hand side of the table, you have luminal digestion, and on the right-hand side of the table, you have membrane digestion. Any starch, any carbohydrate that you're going to consume in the lumen, in the oral cavity, stomach, or intestine, we're going to get amylase, remember, one digestive enzyme, amylase, and it's going to break it. Dextrins, maltotriose, maltose, trehalose, lactose, sucrose, whatever O's there is, basically. Again, in the lumen of the gut, oral cavity, stomach, and intestine. Amylase, one digestive enzyme. Now, after you get into the alpha dextrins and maltotriose and maltose, trehalose, lactose, sucrose, any product, it's going to go to the membrane now. And at the membrane level, you have a special digestive enzyme that is attached to the membrane of the apical cell, the enterocyte. That enzyme, the specific enzyme now, is glucoamylase, which is the upper part of the table. Glucoamylase, isomaltase, sucrase, lactase, trehalase. Now, what does the numbers below it mean? I'll give you an example. If we go to the alpha dextrins, then 5% of the alpha dextrins are digested at the membrane level we're talking about by glucoamylase and 95% by isomaltase. The end product, as the arrow is pointing, is glucose. Done. Second one is maltotriose. Maltotriose, if we're gonna follow the line, 25% is digested by glucoamylase, 50% is by isomaltase, and 25% is by sucrase. The end product again is glucose. Only 3 only 3 of the carbohydrate luminal digestion products, trehalose, lactose, and sucrose, are digested 100% by specific membranous enzyme. For example, sucrose is 100% digested by sucrase. Lactose is 100% digested by lactase. Trehalose is 100% digested by trehalase. The trehalose produce at the end glucose, whereas lactose produces at the end galactose, sucrose produces at the end fructose. These are the 3 end products that we talked about from the carbohydrate digestion. So we have 2 types of digestion. We have luminal digestion and we have membranous digestion. The luminal digestion occur by one enzyme for carbohydrates, and that is amylase, either in the oral cavity, either in the stomach, either in the intestine. That's luminal digestion by amylase. The products, of course, I'm not going to ask about all of these numbers, and you know, you can look it up and see what's the percentage for each of the enzymes that functions on them. But I think trehalose, lactose, and sucrose are very easy. Trehalose, lactose, and sucrose are digested by specific membrane enzymes, membrane-bound enzymes, trehalase, lactase, sucrase. So instead of trehalose, lactose, sucrose, you just add the word ase for the enzyme. It becomes trehalase, lactase, sucrase. Trehalose goes to become, at the end product, as glucose. Lactose becomes galactose, and sucrose becomes fructose. This is basically a summary of the digestion of carbohydrates. Now, after we get the glucose, galactose, and fructose, the end products of carbohydrate digestion, how are we going to absorb it? That's the absorption part now. How are we going to get it inside the enterocyte first? And then after the enterocyte, how are we going to take it from the enterocyte to the blood vessels to go to the body? Because if it stays in the enterocyte, we cannot utilize it. We have to go take it to the blood vessel so the blood can take it to the rest of the body, to pump it, basically. This picture shows how we're going to do that. Very easy. Glucose and galactose requires a carrier that we call SGLT1. SGLT1 carries glucose or galactose to the inside of the enterocyte. This is a picture of the enterocyte. Fructose, on the other hand, requires a carrier that's called GLUT5. GLUT5. When these carriers are present, then they carry glucose, galactose, and fructose to the inside of the enterocyte, as shown in the picture. In the middle of the enterocyte, you have glucose, galactose, and fructose, and then all of these requires GLUT2 to go to the blood vessels. A carrier, a special carrier, that's called GLUT2 to go to the blood vessel, to go to the rest of the body, basically. So we said that you have 2 types of digestion. You have a luminal in the lumen of the gut, oral cavity, stomach, intestine, and you have membranous digestion, which happens by enzymes that are bound to the membrane of the apical membrane of the enterocytes. The end product of carbohydrate digestion, any carbohydrate that's digested in the lumen will work or will be digested by amylase. Done. One enzyme, amylase, in the mouth, in the stomach, in the intestine, wherever it is. It's amylase, only amylase. And it gets you a product. I said three of them, trehalose, lactose, and sucrose are basically what we want to remember because 100% of them are basically digested by specific enzymes. Trehalase, lactase, sucrase. And the end product is going to be glucose, galactose, and fructose. This is the digestion of the carbohydrates. Now, we want to get the end product, which glucose, galactose, and fructose, into the enterocyte first. This is the first step of absorption. Then the second step of absorption is to get it to the bloodstream. So the first step to the inside of the enterocyte, we require a couple of carrier proteins. We call that SGLT1, which carries glucose and galactose, and GLUT5, which carries fructose. Now, glucose, galactose, and fructose are inside the enterocyte. Again, we need another special carrier protein that will carry the glucose, galactose, fructose into the bloodstream to send it to the rest of the cells, to the rest of the body. And this carrier is GLUT2. This is the end of carbohydrates, digestion, and absorption. Now we're going to be talking about the digestion of proteins. And the digestion of proteins is basically, just like carbohydrates, we're going to be talking about the luminal digestion and then the membrane digestion. The end product for the protein is amino acids, basically, because the amino acids are the building blocks for any protein, just like glucose, galactose, and fructose are the building blocks for any carbohydrate. So what happened in the lumen? And again, when I talk about the lumen, I talk about the oral cavity, stomach, and intestine. These are the lumens of the gastrointestinal tract. In the lumen, when you take any piece of steak, again, back to the steak example, you have a couple of groups of enzymes that will break the proteins in the lumen. Endopeptidases and exopeptidases. Endopeptidases are basically enzymes that attacks the internal bonds in the protein. Exopeptidases, on the other hand, attacks the outside bonds. Some of the enzymes that belong to the endopeptidase group, again, enzymes that attacks the inside peptide bonds of the protein, are pepsin, trypsin, chymotrypsin, elastase. Example of the exopeptidases, enzymes which attacks the outside, the external peptide bond, are carboxypeptidase A and carboxypeptidase B. Now, these are the enzymes that will hydrolyze any protein. They will attack it both from the inside and from the outside. And these enzymes are located in the lumens of the gastrointestinal tract. And I know there are no plural for the lumen of the gastrointestinal tract because it's only one tube, the gastrointestinal tract, and because there's only one lumen for that one tube that extends from the mouth to the anus, according to the definition of the GI tract. But to simplify things, we consider the oral cavity is a lumen, the stomach is a lumen, the intestine is a lumen, just to make the point that there are luminal digestion. These endopeptidases are located in these lumens, the oral cavity, stomach, and intestines. And we've talked about pepsin and others or trypsin after pancreatic activation. Now, what happened at the membrane level for protein digestion? This is a picture that shows a couple of enterocytes, and on the apical membrane of these enterocytes, you have a group of enzymes that's called enterokinases. Enterokinases are protein-bound enzymes, membrane protein-bound enzymes. What happens is these enterokinases activates trypsinogen to become trypsin, and when trypsin is present, it starts converting more trypsinogen into trypsin, more chymotrypsinogen into chymotrypsin, more proelastase to elastase, pro-carboxypeptidase A and B to carboxypeptidase A and B. So again, we have two types of digestion for the proteins, one in the lumen, and this happens by two groups of enzymes. We call them endopeptidases and exopeptidases. They attack the external and the internal peptide bonds of the protein in the lumen, in the oral cavity, stomach, and intestine. And after that, when the protein size is reasonable, small enough, it binds to the membrane of the apical membrane of the enterocyte, and there a group of enzymes called enterokinases, which are membrane bound, they start activating trypsinogen to trypsin, which this trypsin starts to convert a number of enzymes to become active so they can hydrolyze the proteins into their final products. So what are the final products of protein? Of course, as I mentioned earlier, it's amino acids. So we have free amino acids, we have di and tripeptides. So sometimes they're two together, sometimes at the most they have three together, three amino acids together. But mostly it's free amino acids, the very basic form. And they go through simple diffusion. They don't need any carriers like the glucose that we talked about. They don't require anything. They only go by simple diffusion, which means that it's a gradient. So you have their concentration outside the enterocyte high, and basically they simply diffuse into the inside of the cell. If they are large proteins, as I mentioned, with regards to the antigens and things like that, large proteins, large size proteins, then penocytosis will take place. And again, this penocytosis, just as a side note, decreases with age. So the older the animal or the individual, the lesser their capabilities of absorbing larger proteins into their cells. Let's take a look at the summary of the protein digestion here. This is a summary slide of protein absorption. Protein in the lumen of the gastrointestinal tract, the oral cavity, and the stomach and the intestine. It's attacked by exo and endopeptidases. When it's digested, it produces amino acids, di and tripeptides, and different kinds of peptides. Free amino acids, peptides, di and tripeptides. As I mentioned, di and tripeptides move to the inside of the cell by simple diffusion. Free amino acids also go through simple diffusion. The peptides, which are more complicated, requires activation of the enterokinases, which are membrane-bound enzymes. They activate a trypsinogen to become trypsin. Once trypsin is present, it starts converting a group of of enzymes to trypsin, to chymotrypsin, and elastase, carboxypeptidase A and B. These five enzymes, they start hydrolyzing the peptides at the membrane level, and then of course the end product will be again free amino acids and di and tripeptides, which will move by simple diffusion to the enterocyte and also to the blood vessels. So simple diffusions, no need for any carriers, no need for energy. Basically, it's a gradient. Their gradient is high on the outside of the enterocyte. They diffuse into the inside of the enterocyte where their gradient is low. So they go from high to low. Lipid digestion. We talked about two nutrients so far. First, carbohydrates, and second, we talked about proteins. We have two types of digestion. One that happens in the lumen of the gut, oral cavity, stomach, and intestine, and one that happens at the level of the apical membrane of the enterocyte by membrane-bound enzymes. They have to be membrane-bound. That's how you're going to get it to the inside of the enterocyte. I've given you a couple of summary slides for the protein digestion and for the carbohydrate digestion, and I gave you a summary slide for the absorption of each of them, carbohydrates and also proteins. Now the third and the last component of nutrients, which is fat. The end products of fat digestion is cholesterol, free fatty acids, monoglycerides, and glycerol. Again, just like the end products for carbohydrates are glucose, galactose, and fructose, just like free amino acids, maybe bi and tripeptides are the end products for protein digestion, the end product for lipids digestion are cholesterol, free fatty acids, monoglycerides, and glycerol. Glycerol, free fatty acids, and monoglycerides get into the lacteal or the lymphatic vessel. They go to the thoracic duct before they end into the venous circulation and then to the rest of the body. Glycerol, on the other hand, goes to the blood vessels directly without any lacteal. So let's see how is this first how they are structured, and then after that how they are digested and absorbed. So the structure of lipids are basically either triglycerides, phospholipids, and cholesterol. The triglycerides have 3 fatty acids and glycerol. Phospholipids have 2 fatty acids and glycerol. Cholesterol have 1 fatty acid and glycerol. And if you remember, we've talked about phospholipid in details when we talked about the secretions of the gallbladder, because this is a very important component of gallbladder secretions. The most important thing about it is it's amphipathic. Amphipathic means it's lipophilic, and hydrophilic means it loves fat and it loves water. What does that mean? It means that it creates a very nice reaction medium, so the enzymes that digest fat can work on these fat droplets very, very, very efficiently. Again, by the function of phospholipids, by the properties of phospholipids because of their amphipathic properties. We will talk about that again in a few minutes. But we have to remember that the structure of lipids are either triglycerides, phospholipids, and cholesterol. Now, what are the functions of lipid? Lipid, I mean, we talked about how bile acids are formed mostly by lipid. Steroid hormones are all formed by lipid. Cell membranes are all formed by lipid. It's also worth mentioning that lipid is a very good source for calories, for energy. The Asian diet, just as an example, Asian diet uh, basically have about 12% of their caloric intake. So Asians take about 12% of their energy from fat, whereas Westerners take about 40% of their caloric source from fats. That's pretty high. That's pretty high. Okay, so so now we will talk about how we're gonna digest fat first, and then how we're gonna absorb it. Okay, so the very first thing we will have is that the different motility patterns in the gastrointestinal tract. We talked about the reservoir. We talked about the chewing in the oral cavity and the swallowing in the pharynx. And we talked about the two motility patterns in the stomach as the reservoir and the antral pump. Then we have the gastric emptying. Then we have the MMCs in the small intestines, mixing and short peristaltic movement and propulsive movements. All of these create fat droplets. So you're not going to take the fat in the piece of steak as is. The different motility patterns will break it into fat droplets. This fat droplet, this fat droplet will be easier for enzymes that digest fat to work on it. So the fat droplet gets hydrolyzed by lipase, and then it will be emulsified, meaning that it will have an electrical charge added to it. Remember the phospholipids and the hydrophilic affinity for that. Lipophilic and hydrophilic, the amphipathic properties of phospholipids, which makes it more and more available for lipase and phospholipases to work on it. So we then emulsify it, and then we do the mycel formation, which will basically is a water-soluble molecule that will get into the enterocyte. So there are a number of steps for fat digestion. Gut motility, hydrolysis, and emulsification all happen in the luminal digestion. The mycel formation also happen in the lumen, but at the level of the apical membrane of the enterocyte, that's when the micelle basically uh, uh, give gives up its content for the enterocyte to take them, and that's the part of the absorption of fat. So let's see how these things work. First, I want you to remember that gut motility also participates in fat digestion. It forms fat droplets. After that, hydrolysis takes place. Lipase starts to work, and then emulsification, adding an electric charge by bile salts and phospholipids, and and then the end stage is a micelle, formation. So let's see how these things work. Hydrolysis. So we learned that the motility pattern will basically form the fat droplet. The next step is hydrolysis of lipids. Well, how does hydrolysis work? How the lipid is going to be water soluble? Well, you have three steps to do that. First step is by the enzyme lipase, which is activated by colipase. Colipase activates lipase, and lipase starts digesting the fat droplets. Now, there's also There's also phospholipase A, which requires bile acids for activity, meaning that you have to have bile acids in the medium that will attach to the fat droplets, and that's when phospholipase A2 starts working. Cholesterol ester hydrolase is a third enzyme that you will utilize to basically digest the fat droplets. So first of all, you go with the motility patterns to form fat droplets. After forming the fat droplets, you have three enzymes that will work: lipase, phospholipase A2, and then cholesterol ester hydrolase. Now, how does that happen? We showed this picture before about the uh, shape and the structure of a bile acid. The upper picture showed the chemical structure, and the lower picture shows a cartoon drawing of how does the bile salt or the bile acid look like. So you have a non-polar side, a side that does not like electrical activity or does not like water or fat, it just gets away from them. And then you have the carboxyl group, which basically are polar. They are reactive. They can work in a liquid medium. So this is basically the first structure for the bile salt. Now, I said that the motility patterns form fat droplets. After that, what happens, and we've seen this picture before, is that bile salts starts to attach, start to surround these fat droplets, which will give the chance for lipase to start working on them, to break them, to digest them, to emulsify them, along with phospholipid, which prevents their aggregation. The more phospholipid you have, the better the fat digestion process goes, because you will have the fat droplets isolated by themselves, which will give lipase a better chance to work on them. That's very important. So you have bile salts and phospholipids to do this job. Bile salts create an electric charge on the fat droplets so lipase can work on them to digestion, and phospholipid prevents their aggregation. That's very important. Phospholipid isolate them, and so lipase and phospholipase, A2, and cholesterol ester hydrolase will also work on them, less specifically, but still work on them. This is the emulsification process. The third process is the micelle formation, and the micelle formation is the end product. So basically, all what you have is a monoglyceride or a fatty acid attached to the bile salt. Of course, the bile salt is not going to be absorbed to the enterocyte. Only the fatty acids will be going to the bile acids and fatty acids and monoglycerides, as well as the cholesterol will be going into the enterocyte, whereas the bile salt is going to be reabsorbed and going back to the gallbladder after the blood circulation. This is how these are the end products of fat digestion: cholesterol, fatty acids, and monoglyceride, then glycerol. Glycerol goes into the bloodstream immediately, straight to the bloodstream. No carriers, no special transport, binding proteins, no nothing. Goes simple diffusion to the bloodstream. Cholesterol, fatty acids, and monoglycerides, on the other hand, leave the micelle, the micelle and gets inside the enterocyte by fatty acids binding proteins, FABP, fatty acids binding proteins. They carry them to the inside. This is membrane digestion now. You take them and you absorb them. I'm sorry, not membrane digestion, but an absorption of the fat end product, fat digestion end product, cholesterol, fatty acids, and monoglycerides. They are absorbed into the inside of the enterocyte by fatty acids binding proteins. After that, a number of chemical changes happen to repackage them into what we call chylomicron. So cholesterol gets into the smooth endoplasmic reticulum, fatty acids with colipase A, monoglycerides, all of them with phospholipids, phospholipids goes into form one molecule called chylomicron. And so the chylomicron is the molecule that gets into the lymphatic system, the lacteal first. The lacteal is the lymphatic system, goes after that to the thoracic duct and then to the venous circulation. So what are the components of the chylomicron? How did chylomicron form? There is a protein that's called apolipoprotein or apoprotein. That's 1%. There's cholesterol, 2%. Triglycerides, which is basically from fatty acids and monoglycerides and diglycerides, forms triglycerides, 90% of the chylomicron. And then phospholipids, 7%. All of them form chylomicron, and the chylomicron basically gets into the lacteal or the lymphatic blood vessel. Glycerol, on the other hand, goes straight to the blood. So, steps of lipid absorption is basically the micelle formation and then the chylomicron formation. Micelle formation gets you attached to a bile salt, bile acid, and then end up like a cholesterol, fatty acid, or monoglyceride. After that, you have fatty acids binding proteins to get you inside the enterocyte, and after that, the formation of the chylomicron by apolipoprotein, cholesterol, triglycerides, and phospholipids. All of them form the chylomicron, different percentages. For the highest is 90% triglycerides, basically. And this chylomicron gets into the lacteal. They do not go to the blood circulation first. They go to the lymphatic circulation. Lacteal is the lymphatic vessel. So chylomicrons goes to the lacteals through exocytosis, and glycerol, as I mentioned, by simple diffusion goes to the blood vessel directly. This is again a picture to remind you where the lacteal is, which is in the center of this villi, and beside it from either side is the arteriole and the veniole, which is the vein and the artery for that. Of course, the black line shows the nerves there, the vagal efferents and the enteric nerves that gets into the villi. Now, one last thing about fat digestion, and that is cholesterol. 95% is reabsorbed and 5% goes into the feces, the cholesterol. Why did I choose cholesterol? Because there's a lot of problems related to this molecule, and I'll mention it in a second. So, cholesterol is basically utilized and used for bile synthesis, as I mentioned before in the secretion section. 95% is reabsorbed and 5% goes into the feces. And I also mentioned this number. In the blood vessel, you have two types of proteins that carry cholesterol. Now here's the most important thing, is you have high density lipoproteins and low density lipoproteins. The high density lipoproteins are pretty good. They carry the cholesterol and they move it. They carry it and they move it. So you can utilize it, you can use it for bile acid formation, for anything else. The low density lipoproteins, LDLs, are bad. They keep it, they aggregate it, they make aggregations out of this a molecule. And this is bad when this aggregation take place in the lumen of the blood vessel, as this picture will show you. This picture shows high density lipoproteins and low density lipoproteins carrying cholesterol, which is the small circle on the left hand side of the picture. The green is basically the high density lipoproteins, which are the good ones, which basically move the cholesterol from the blood vessels, take it to the tissues, utilize it, etc., synthesize stuff from it, like bile, etc. The LDL, they don't. They make aggregation. They accumulate it together. They make aggregations of it, just like the right-hand picture shows, the yellow aggregations. That's what we call atherosclerosis. That causes lots and lots of cardiovascular problems because the lumen of the blood vessel becomes pretty narrow, and therefore blood cannot move or will not move freely in the blood vessel, which will lead to strokes and many other problems. This is another picture to show you this example. On the left-hand picture, it shows the atherosclerosis, which is basically the bad protein. The LDL, which is the low density lipoprotein, aggregates cholesterol in the lumen of the blood vessel and makes it hard for the blood to move freely in the lumen of the blood vessel in the left hand side picture, causing again problems, as I mentioned, strokes and things like that. Arteriosclerosis, on the other hand, is a problem that happens with old age mostly, but it happens at any age, but mostly in older animals or individuals. And basically, it's a calcification of some of the layers of the wall of the blood vessel. And when this calcification takes place, it narrows. It makes the blood vessel hard to contract. And when the blood vessel is hard to contract, it's not elastic enough. And that's why the blood basically does not move freely in that blood vessel. And again, you will have problems such as stroke and things like that. So this brings us to the end of the digestion and secretion story. And in summary, we have two types of digestion. We have a luminal that happens in the lumen, and membranous that happens at the apical membrane of the enterocyte. very easily memorized by first one enzyme for carbohydrates, that's amylase, and a number of three specific enzymes for carbohydrates, trehalase, lactase, and sucrase. The end product is glucose, fructose, galactose. We need carrier proteins to get them into the cell. That's SGL2 and SGLT1 and GLUT5, and then from the enterocyte to the blood vessels, we have GLUT2. For the proteins, amino acids is the end product. We have in the lumen endopeptidases and exopeptidases that will basically digest the proteins into free amino acids, dipeptides, and tripeptides, and also other complicated peptides. After that comes the membrane-bound group of proteases, which we call enterokinases, which converts trypsinogen to trypsin, which as a result will convert another 5 different enzymes to make all the proteins become free amino acids, di and tripeptides, and then they get to the cell by simple diffusion. After that comes the fat, and fats you have the luminal digestion by lipase, which is in the oral cavity, stomach, and intestine. And the end product is going to be cholesterol, fatty acids, monoglycerides, and glycerol. This, after that, takes place. The digestion happens in a number of steps. First, formation of a fat droplet, hydrolysis, and then lipase starts to work, and then emulsification, bile salts and bile acids, phospholipids will start working also, and 3 different enzymes will digest that: pancreatic lipase, phospholipase A2, and cholesterol ester hydrolase. Then after that, the micelle formation. Micelle formation basically gets the material into the apical membrane, gets the cholesterol, fatty acids, and monoglycerides to the apical membrane level, and then fatty acids binding proteins takes it inside the enterocyte. Once these are inside the enterocytes, they are repackaged and they form what we call chylomicrons. Chylomicrons goes into the lacteal and goes to the thoracic duct and then to the venous circulation. Whereas glycerol, which is the 4th end product of fat digestion, goes inside the enterocyte and then to the blood vessels freely. Simple diffusion. Next lecture will be the last lecture, and in that lecture I will be talking about fermentation in both ruminants and also in other animals.